Hi, everybody. This is a review recording for our exam, which is the third exam, and it's dedicated to the PowerPoint chapter. It's actually chapter 9 in the Welsh Hall textbook, as well as this chapter 9 in the PowerPoint series that I prefer to use. And it's all about the muscles. So, yeah, and basically, you have to know about the muscle fiber. The key issues are things like this: the the way that that signal is generated. And so if you look at the muscle fiber, once you break it all down, okay, and you get down to the level of the fiber, surrounding the fiber is some connective tissue called endomysium. If you peel that away, you actually see what's called the sarcolemma. The sarcolemma is it's a fancy name for a muscle cell's plasma membrane. Lemma means membrane, or and we're looking at that a little bit when we look at nerves as well. So that sarcolemma is able to generate a signal that goes deep within the muscles. It does so because these sort of specialized extensions, infoldings, invaginations, and any number of words. Look at those animations. They're called T or transverse tubules because they go transversely or at right angles or at 90 degrees to the long axis of that fiber. And you can see them, they envelop those fibrils that are within inside. That's how the signal gets deep inside. And because those T tubules have intimate contact, they're right next to those well like structures we call terminal cisterns that are part of the sarcoplasmic reticulum, which is just another kind of smooth endoplasmic reticulum, except those enlargements on either side of those T tubules throughout that muscle fiber contain a ton of calcium ions and voltage gated channels. And as the signal goes through, it flips the switch and outpours the calcium, which is being held within the cell itself. And effectively it's being held within the sarcoplasmic reticulum itself, and it floods the rest of the cell. Because those are basically like opening a door, that electrical signal. That's what meant by a voltage gated channel. It opens the gate, and we open our gate here, and the dog will shoot out. In this case, that electrical signal is allowing calcium to shoot out. So that's really important. And what it's doing, I mean, this is the basis of what's called excitation contraction coupling. That's marrying the electrical activity, the signal, the action potential, with mechanical activity, the contraction. And so, how is it done? It's done through that combination of the electrical activity going through the T tubules, flipping that switch, releasing the calcium, and it enters the cell, and it generates mechanical activity. Because remember what the calcium does. Down the road, once it gets in there, it's going to attach to this interesting molecule called troponin, a neat protein we call a regulatory protein. It's going to change its shape. And once it changes its shape, it's going to allow that tropomyosin, that thread like protein that envelops that interior of those fibrils. And basically, what it's doing, it's deep inside the inside the sarcomere within those fibrils, and it's covering those active sites. The active sites are on that are covered are the ones that are on the actin filaments. Technically, we call them myosin binding sites, but just know that those active sites are on the actin filaments, and they're being covered by the tropomyosin. And as soon as that calcium comes in, it reacts, bonds to, interacts with the troponin molecule, and basically causes the tropomyosin to shift and expose those heads. And they're in such close proximity to the myosin heads that are already loaded with energy that it causes the contraction. That's basically excitation contraction coupling. And what actually causes the muscle to shorten are the cross bridges that form between the actin that's now exposed, those strands, and then the myosin heads, which are already precoxed, and they're going to fire and pull, and the actins are going to pull the sarcomere boundaries, the Z discs, closer to each other. That's really the crux of what happens inside. So the actin filaments, I always call them like a double string of pearls. That's where the active sites are located. The myosin, I mean, they just look to me, they look like a Twizzler that you cocked up and sort of squeezed or flattened one end of it to make that head where it's going to attach. And what do? I mean, the head basically, it's like pulling it back to load it, and then boom, it'll fire. Just like any number of toys we had when we were kids had that kind of a thing. You had a rubber band and you pull something back, and boom, there it went. So that's the whole idea. That the fact that that protein myosin moves makes it a motor protein by definition. So that's where you are. And again, actin is thin, myosin is thick. Again, the transverse tubules are where basically the action potential, the electrical impulse travel. Okay, that's effectively they're part of the sarcolemma. They're actually part of the plasma membrane, where that sarcoplasmic reticulum, where the calcium is stored in those terminal cisterns. That's really just that. Now we're sort of in the interior of the cell. So now that sarcoplasmic reticulum is basically a variation of smooth endoplasmic reticulum. There's plenty of glycogen in the cell for energy. There's plenty of oxygen available in the cell, but it's being held in a substance called myoglobin. Remember, hemoglobin is in the bloodstream. That's for the rest of your body. Myoglobin is similar, but certainly not the same. Different molecule entirely. But what it does is it releases oxygen when it's needed to generate ATP. And that's what the glycogen is for. That's stored carbohydrates that can be basically utilized in the presence of oxygen to make more ATP. And the skeletal muscle is such an oddball. We call it multinuclear. It's like a fusion of those protein rods we call the fibrils and the filaments inside, and all the different connective tissues, that spring load stuff, the titin, uh, all of the actins and myosins, all of that stuff that's in there basically are like multiple cells that have fused. So effectively, it's multinuclear. It's very unique. You can't make any more of them. We're born with what we have. Yes, they can get bigger. We can make them hypertrophy. And so there will be, I mean, here's the answer key: tropomyosin, the thread like protein, troponin, okay, that plug like protein, ATP, the energy source, calcium, the stuff that basically is released and really starts this magical process of effectively it's released by the excitation and then generates the contraction. So calcium's a big player. It's stored in the terminal cisternae. It's the one that bonds with the troponin. Oh, and oh, by the way, calcium from outside the cell is important too, particularly because it has to be near the nerve by the motor end plate, because that allows the synaptic vesicles. So again, part of this, the synaptic vesicles to release their acetylcholine. That's such a big deal that calcium is all over the place. And lastly, acetylcholine. So again, I'm going to have a series of questions for this. Again, it's a modified matching: tropomyosin, troponin, ATP, calcium, and acetylcholine. I can't give you any more than that. So effectively, just to get the nerve to fire, we need calcium. Just to get the muscle to fire, we have to release calcium. And effectively, then we have to basically take that calcium and pump it back in. So we have to put a lot of energy. We need more energy to detach those heads once they fired and effectively leave them cocked. That's the whole idea. I went over this stuff again and again and again, and you must too to understand it. So the motor end plate is different. We've been talking about with calcium voltage gated channels. This is a ligand gated channel. It's a chemical. The chemical is acetylcholine that opens the gate. I mean, gated channel means you just need something to throw the door open. Okay. Because typically the resting position is closed. And if it wasn't, I would tell you about it. So if it's voltage, it's an electrical current. It's flipping a switch. If it's a chemical, the chemical has to act to throw that gate open. We call that chemical a ligand. You insert the term chemical, but it happens to say ligand gated the way I'm using it on the answers on this test. I told you about the Z discs, how important they are. I told you about the acetylcholine. The big deal with acetylcholine is it's broken apart very, very rapidly. That's why basically that whole idea of frequency keeps spitting out the acetylcholine, and as long as that's being spit out and an action potential is maintained, basically we're going to be pushing out as much calcium as possible. That's the whole idea. Calcium gets broken down. Certainly, remember the three ways. Some of it diffuses. Some of it is broken down directly by an enzyme called acetylcholinesterase, and even some of that acetic acid diffuses. And lastly, the choline is sucked back up. It's reutilized. It's pumped back in. We call that reuptake of that. The big deal and why it moves so fast is because the enzyme is there and it's always there. We all have within that synaptic cleft, that neuromuscular junction, this enzyme called acetylcholinesterase. That's so pivotal. That's what takes it away. So muscles fire fast and they reset fast. Think of it that way. And it's because of that enzyme. And that's the big problem with that enzyme. It's so vital. If we do something to it, I mean, if you took it away, theoretically, the muscle would constantly fire. And that's exactly what happens in nerve gases and chemical weapons like sarin or VX. They are what are called acetylcholinesterase inhibitors. Those inhibitors are extraordinarily dangerous, particularly when they're weaponized. So they effectively are going to inhale this poison chemical. Effectively, it's nerve gas, but effectively that's chemical warfare. Yes, with tiny amounts, there's different things that we utilize these things for specific conditions. But it's the whole idea that this is really a foundation to understand how neurotransmitters, and you're gonna learn more about that when we get into the nerve system. Neurotransmitters, yes, they diffuse away, but a lot of the times it's based on enzymes that break them down. Okay, so breakdown, metabolism, that's one way. That's what the acetylcholinesterase does. And lastly, reuptake. And sometimes the mechanisms, they just suck it back up and recycle it, repackage it in a synaptic vesicle and get it to spit out again. Remember A bands and I bands. I'm just thinking about that. There's no A, there's an A in dark and there's an I in light. Okay, remembering that. The A band is where they overlap. That's why it starts, the thin and thick filaments. It's light because they don't overlap. That is there. And I commented on that a little bit before. I made a point about it. That's when I looked at those electromyograms. And that's what I meant by frequency. And that's what the acetylcholinesterase kind of lets you do. You keep firing away. And because it resets very quickly, and then you can pump out more calcium out of that. If you pump it to the order of 50 to 100 times a second, you're going to reach something called tetany. You're going to induce a spasm. We use that in physical therapy. That's why it's important. That's very, very rapid stimuli. Remember how I looked at it in class? 10 of those a second, it gives it time to relax completely. 20 of those a second, we start to get that staircasing if we start to increase the frequency a little bit, because not all the calcium gets pumped back in. If we get to 30 a second, we get that unfused tetany. And if you get to between 50 to 100 a second, then you get pure tetany, the complete spasm. And again, this is done in a laboratory. Particularly, it's done in physical therapy. We use it for rehabilitative means to try to treat things like that. So, what actually does the acetylcholine do at the motor end plate by way of those ligand gated channels? It basically opens the door. It's an interesting door because sodium goes in and potassium goes out. 3 sodiums go in and 2 potassiums go out. Because of that, the interior around the motor end plate, and if it's sufficient, the entire sarcolemma, the entire membrane, will basically become excited. We call it, if it spreads far enough, an action potential. So remember, all it's doing, it's opening up the doorway. Basically, 3 sodiums can get in, potassium is bigger, 2 potassiums can escape. The net effect is to make it more positive or less negative. In that motor end plate, we call those end plate potentials. And if we do it frequently enough in a small area, what's going to happen is going to be a big enough change to generate that action potential where it's going to spread and contact those other channels, which happens to be voltage gated sodium and then voltage gated potassium channels. That's how it works. We did a little bit more on those energetics when I talked a little bit about aerobics and anaerobic. The big take home point is we very rarely need these extra sources of energy. We very rarely do activity with muscles that involve that much. We do it on such short bursts. We've got about enough ATP available for 4 to 6 seconds. After that, once that's used up, we have another high energy molecule that can be used. It's called creatine phosphate. It's kind of like ATP that it has a high energy phosphate. The big P in ATP stands for a phosphate. We can swap it out. Once we've used up the one in ATP, we can grab another one from the creatine phosphate. That'll last under 10 seconds. When they're gone, if you had to continue doing this extremely vigorous muscular activity, okay, at that point, we start to build up lactic acid. We don't have the muscles contracting so much by this time. Oxygen can't get in. Without oxygen and without blood supplies that nourish it and bring more supplies of glucose in, it's going to become anaerobic, and it's going to use anything it possibly can to burn. The trouble is it's going to build up a lot of waste products like lactic acid, which will get to the bloodstream and cause problems, and a whole bunch of waste products, particularly charged particles, that eventually are going to damage the muscle. And at first, just cause it to spasm and get so painful, you just have to stop. And effectively, when you stop, you have to replenish. You have to replenish oxygen so you can counteract the effects of the lactic acid and eventually get the muscle to work again so you can get rid of those waste products. They have a fancy term, that EPOC. I always said catching your breath. You basically have an oxygen debt, would be a good way to express it. And that really was much more of a traditional term. If you want to turn the exercise into an aerobic exercise, then you have to marry your breathing and your heart rate. As your heart rate goes faster as you use the muscle, and you're using big muscles like your arms and your thighs, either one or the other, or preferably both, what's going to happen is you're going to marry your breathing to the heart rate. So you bring in the oxygen and get rid of the carbon dioxide, and effectively you can scientifically stress whatever structures to build up circulation, reserves, and endurance, and particularly the heart. That's what cardiac aerobic conditioning, or what we call in the gym cardio. And say, hey, I'm working on my cardio. That's when you're on the treadmill, you're doing that type of stuff. Really important. So all of those are important things. Again, remember where the binding sites are. They're on the myosin, but they're already exposed. They're on the myosin heads, and on the actin, they're the ones that are covered by the tropomyosin. And when all this is said and done, while this is going on, that cell is sodiums and potassiums are flying around all over the place. Those, because in nerves and in muscles, and we're talking about muscles here, what happens is those sodium potassium pumps get extraordinarily busy then, and they're constantly resetting the chemical balance inside the cell. Remember what I told you? Electrical events come first. Then we have to fix the chemicals. Even though we've gone, we've depolarized, it's fired and the cells reset electrically. We have to reset the chemical balance so it can keep doing it again and again. That's what the sodium potassium pumps do. They're not the same as the sodium potassium channels, whether they're ligand gated or voltage gated. Those pumps are all about active transport. So we're generating more energy just to reset that system so we can use it again and again. That's really important. And because of that, we need a tremendous amount of energy. To do this, we have to reset those pumps. We have to, in order to detach the muscle so it can keep firing or fire again, we have to use ATP. When we stop, we have to have ATP to push the calcium back into the wells. We have to have ATP for the nerve to fire. And because of that, okay, and the fact that the ATP has precocked those, that's where the phenomenon of rigor mortis comes from. Rigor mortis persists because once somebody dies, those muscles are still ready to fire. As soon as the muscle starts to deteriorate a little bit, the calcium spills out, being contained inside those wells. And that's what causes the muscles to become rigid. It stays rigid, depending on the conditions, let's say, somewhere between, 4 to 6 hours, all the way up to maybe 24 hours. And what causes it to deteriorate, okay, eventually, okay, is that the muscles are just going to denature. The proteins, it's basically going to start to rot. for lack of a better thing. If you want to think of a deer, roadkill looks like the deer's real rigid sometimes, and then starting to kind of go all over the place. Yuck, that type of thing. The reason it stays rigid until the proteins start to break down completely is because we don't have any more ATP. Something that's died can no longer generate ATP. And then we have a few questions about smooth muscles. And remember, the difference is that why we call them smooth is we can't see any striations because we have either 12 or 13 to 1 thin to thick filaments. Skeletal muscle is 2 to 1. And it's totally different. It's it relies on extracellular calcium ions. It's a single cell that's basically got one nucleus. It's got a different shape. It's not cylindrical. It's that spindle shape, narrower in the edges, wider in the middle, so it puffs when it contracts. Okay. It doesn't have transverse tubules to conduct the signal. It has those caveolae. There's really just sort of, again, like a little indentation, it's sort of like an area that flows, that kind of looks like it takes the membrane inward a little bit, where the calcium is going to get in. The calcium is coming from the outside anyway. There's a little bit on the inside. It doesn't have troponin. It doesn't have a neuromuscular junction. Okay. It doesn't have any of those. It's got those varicosities. Those are just those swellings in the nerve. They have the synaptic vesicles. They have mitochondria. They release the neurotransmitter, but it's not a neuromuscular junction. It's different. It doesn't have the T tubules. It has those little indentations, caveolae, and those are where the voltage gated channels are. So as the signal comes through by those caveolae, boom, it opens them up. Calcium from outside the cell flows in. That's why it's so important. And it even allows some of the calcium that's in the sarcoplasmic reticulum in the cell to come out. But that's not a big player. The calcium bonds to something called calmodulin, changes its shape, and that's what creates the contraction. It contracts in a spiral manner. That's because of the way that those intermediate filaments are in that lattice formation and they're anchored by those dense bodies. The dense bodies are kind of like the Z discs that you see in the sarcomere because it doesn't have any Z discs. It's that those sort of takes the place and why it contracts in that sort of unique spiral manner and kind of puffs up in the middle. Okay. So the big deal is the calcium is from the outside. That's really important. Same with the heart. And the dense bodies are like the Z discs. Okay. Smooth muscle is certainly not as powerful, but it has tremendous endurance. It has a very specific arrangement. Remember, I went over that. We have, remember, kind of like pushing the toothpaste out, the peristalsis. Typically, it has a sheet of circular muscle or sheets of circular muscle around the tube. And then around the circular, it has longitudinal muscles. So it can squeeze and push and squeeze and push. So it's not a random arrangement. It's a very organized arrangement, sometimes even more organized than that. And I think, again, that's all part of this unbelievable picture. Don't forget the importance of all those other things, the troponin, where the tropomyosin plays a role when you're dealing with skeletal muscles. There aren't as many questions on smooth as there are on skeletal, but you do need to know about both. And remember, the critical piece when we see depolarization, the big deal about depolarization is it's because remember when the action potential starts with depolarization and then reset with repolarization. Depolarization is the vast flowing in of sodium. Repolarization is that vast, big time flowing out of potassium. It's because of that those sodium potassium pumps have to quickly reset it. When the sodium rushes in, when you hit an action potential all over the place, a ton of sodium comes in and changes the polarity. It goes from positive on the outside, now it's positive on the inside. Rapidly thereafter, the sodium gates open. Those sodium gated channels open. The sodiums flow out. That brings it back, rapidly toward its resting potential. But now you have really effectively flip flopped. The sodium's too much on the inside, the potassium's too much on the outside. That's what the pumps do, is they fix that right away. And I think that's everything I think I got to cover here. So listen to these, form questions, talk to me. We've got plenty of time to do this. We have, from way I'm doing this on Thursday night to 6, so you should have, you got more than a week and a half to take care of this thing. So let me know, and that concludes this presentation.